SARS-CoV-2, as you all know, it is a single-stranded positive RNA with 4 major structural proteins, the most important of which is the S protein, responsible for the crown-like appearance molecularly of the virus. We have developed lessons from previous coronavirus vaccines that have been developed towards the SARS-CoV-1 and the Middle East Respiratory Syndrome virus, or the MERS virus. These are the two cousins of the current SARS-CoV-2 that have circulated in the early 2000s in the case of SARS, and in 2014 in the case of MERS. There is a 30% homology in the spike protein between the spike protein of the SARS-CoV-2 and the MERS spike protein. There's also an 80% homology in the sequence of the spike protein on the surface of SARS-CoV-2 with SARS-CoV-1. In the phase 1 human trials for the vaccine for SARS and MERS, we have detected broadly neutralizing antibodies in vivo in the recruited healthy volunteers. The development for the MERS vaccine still continues, but the SARS vaccine has stopped. This is the crystal structure of the spike protein on the surface of the SARS-CoV-2. It is important to note that the spike protein in the SARS-CoV-2, or the COVID-19, is responsible for the viral entry of the SARS-CoV-2 upon engagement with the human host cell. So therefore, it is important to understand the specific sequence for the S protein, as well as the exact crystal structure for that protein as we design the vaccine targeting specific amino acid stretches of the S protein. It is a trimeric fusion protein. It undergoes substantial structural rearrangement to fuse with the viral membrane, with the host cell membrane. A sequential process of cleavage takes place in 2 subunits of the spike protein upon viral entry. The criteria, then this patient is unblinded to see if he or she received the placebo or the real vaccine. The summary of the phase 3 results so far shows that the mRNA vaccine encodes for the S2P in a lipid nanoparticle designed for delivery for the antigen-presenting cells of the lymph node. The preclinical data have demonstrated induction of neutralizing antibodies. The interim data from phase 1 indicate that 100 microgram dose of the vaccine is the best tolerated dose across age strata. Induction as well of neutralizing antibodies occurred, and most importantly, phase 1 and 2 and 3 so far showed that the antibodies generated in the recipients are higher than the level of antibodies in the convalescent sera. The neutralizing antibodies in the convalescent sera was used as a comparator to the level of the antibodies developed by the recipients of the investigational vaccine. And it was proven that the level of antibodies in the recipients of the investigational vaccines were higher than those in the bodies of or in the samples of the convalescent sera, which is a good metric of the efficacy of these vaccines. The summary is that the vaccines that are currently in phase 3 of the clinical trials has generally showed appropriate safety and efficacy measures. However, we do need multiple approved vaccines, and I expect by this time next year, we may have more than 2 or 3 approved vaccines worldwide, and even beyond that for years to come. There are, however, many challenges regarding the storage of these vaccines, the temperature, the delivery, how to manage giving. In Egypt, for example, 100 million Egyptians requires, in this case, 200 million shots, making sure that these vaccines are stored in the appropriate temperatures. They are stored in the logistics, the management of the logistics in this case would happen in the correct way, and making sure that the healthy recipients of these vaccines are receiving the vaccines in the exact timelines, 21 days post the first shot. This will be a huge challenge. And in the US, the preparatory steps for storing and administering these vaccines are already underway, and it is indeed one of the main challenges facing all the world once these vaccines are developed and approved, how to administer them efficiently. Thank you for giving me the opportunity to discuss this matter, and I will be glad to take any questions.